The introduction agenda is I'll talk a little bit about the tools to increase bio-relevance of assays, why 3D cell models, the plates and the samples for doing 3D cell models, iPSC-derived cell lines, and then high-content screening tools, an overview of high-content, the MetaXpress microconfocal, which is our latest product, and the software that drives it. So the use of 3D models is expanding. It's quite hot right now. People are trying to use it for translational research. People have been doing 2D assays for quite a long time, and there's this transition to 3D assays. People are moving to it mostly because there's a belief that it's more effective of in vivo cell morphology, gene expression, differentiation. There's data to back that up. And more closely recapitulates in vivo responses to drug treatment, particularly drug resistance, with all of this being the goal of improving predictivity and relevance of cell-based assays and moving those into a screening environment. There are some challenges of using imaging, these more biologically relevant assays. Scale, how do you acquire 3D images across multiple treatment wells in this case? How do you analyze that data across multiple treatments, either in 2D or 3D? Of a 3D sample, what's the right biologically relevant cell models, and how do you capture the structural detail and accurate marker localization in complex models, such as spheroids and or tumor models, whole organisms, hydrogel or extracellular matrix assays, or co-culture assays. So we'll touch on some of these, trying to overcome some of these barriers in this webinar. So first of all, what do you put your cells in? So we're going to be featuring Corning's ultra-low attachment, unique round well-bottom plates. They are fit into anti-SPS footprint detectors and they have them in 96 and 384 well. We've been publishing quite a bit with these plates ourselves. They're clear bottom. You can visualize them for imaging, and I'll show you can reproducibly form spheroids at the bottom of these plates. We've developed a very simplified workflow for culture and compound addition. You can add your cells to these wells as you would normally, and after 1 or 2 days, you've got a nice spheroids formed, and there's a time-lapse movie showing that. And there's a panel on the right showing where this is the time-lapse data over, just as a montage. And then what's nice is you can just, and Oksana will show you, can just add your dyes to these wells and do your experiment. We found that you can make very reproducible spheroids depending on, and you can control the size of the spheroids by the number of cells you add to the well and the time you incubate those cells. This is a 96 well taken, 96 well plate taken at 10x, and we get about a 10% variation in spheroid size, which is quite reproducible for these assays. And we can, you can see we're automatically acquiring that data across the 96 wells. A number of cell lines have been validated for spheroid formation, demonstrating broad utility of using these plates. And some of those plates, cell types are in the table below. And at the top, you see the AC12 HeLa and A549 seeded at 5,000 a well. And then these are breast cancer cells, MCF7s, and DU145 cells, all illustrating that you can form the spheroids, and then much more have been done. And in our hands, everything we've tried really has formed the spheroid. You can also do co-culture assays. We are not going to be illustrating those in the webinar, but just want to highlight that you can bring together a more relevant tumor microenvironment with adding stromal and immune cells. And here's some images showing A549 cells with fibroblasts and PBMCs added to the co-culture system, so that you can have a more complex biology to be able to do your assay in. The other, so we talked a little bit about cancer cells and doing those in cancer cells, but if you don't doing cancer biology and you want to move into a more biologically relevant system, you can, and you want to do this in a sort of human environment, iPSC-derived cell lines are a nice technology. And we've been working for a long time with Cellular Dynamics International, who get human donor samples, induce them to be stem cells, and then have a whole slew of products from cardiomyocytes to neurons to astrocytes that you can then develop assays on. And those are available as cryopreserved cells, and they provide it globally to people who want to do that. And we will be highlighting a couple of these cell lines in this webinar. Why iPSC? Well, getting to the right biology is key. Cardiomyocytes have the right amount of electrical activity. They have cell signaling. They have mechanical contraction, and you'll see some of that in the webinar later. We also are using iCell hepatocytes, would have the right number of the right metabolic dependent toxicity, and they show phospholipidosis. So we want to use functional cell types to be able to get to translatable answers. On the imaging side, so we provide an automated imaging tool, and how would you do these experiments if you didn't have such a tool? So with a conventional microscope, you'd have your sample on a microscope, you could take your pictures, and then the question is, how do you make your measurements? Your labs and hands-on time will be typically is hours. With the process with the MetaXpress microsystem is to convert, to speed that up. You can use a variety of tools. You put them in the system, collect a variety of data, and try to make sense of that data in minutes rather than in hours. With the molecular devices products, hundreds of automated applications have been validated, and here is just a sampling of them. And we like to say we take the customer from apoptosis to zebrafish. So angiogenesis, apoptosis, membrane analysis. You're going to see a lot about spheroid analysis and stem cell biology, but people are using our technology for a variety of applications. A complete solution for imaging includes acquiring the images, identifying those objects through image analysis, collecting multi-parametric data output, and then making sense of all the numbers that you're collecting. So here's highlighted some images taken by the system, all around doing these with more biologically relevant assays, tissues, spheroids, hydrogel assays, co-culture assays, some whole organisms, and antibody binding in a background of the antibodies not removed. We like to provide a product that allows you to choose the mode that's right for your assay. We have tools for wide field imaging, which is ideal for flat biology. You might be looking for some upregulation of some gene. You might be trying to do a time-lapse assay looking at some endocytosis function, which is shown on the right. If you have a thick sample, like the spheroids we're going to be talking about, or cells in a hydrogel, then confocal imaging provides superior background rejection and is great for thick samples. And that's what we're going to be talking about using today, because we're mostly talking about the spheroids. Just because you're moving to confocal doesn't mean you can't do live cell assays. So at the top is a sensor that gets very bright in the nucleus and shows a puncture pattern when the cells are in S phase. This movie is taken over 16 hours in confocal mode, and you can see some of these cells are dividing. Right here, these cells are dividing. Cells are going through. You see them enter S phase, and then and here you can see these cells are dividing. So just because it's confocal doesn't mean that it's not, it's killing the cells. So you can do that over a very long time course. And there's a different example shown at the bottom that I'm not going to highlight. But again, a long time lapse, cells are going through their normal cell cycle events. So a complete imaging workflow from Method Devices includes an MetaXpress system at the top. It's driven by MetaXpress software, and MetaXpress allows us to do the analysis, and I'll talk about that next. It's tied together by a data management solution so that you do not need to export the images or the measurements to make sensitive data, and we have a data mining software for that. On the confocal side, we deployed something called Agile Optics technology that allows you to choose the mode that's right for your research. It includes a bright solid-state light engine, so that allows you to do those, get enough light to the sample to be able to do confocal imaging either in 2D or 3D. We use a proprietary spinning disk technology, which I'll talk about in a minute, that has a variety of geometries to optimize your detector, your ATS platform for the biology you're going to be looking at, and a high sensitivity CMOS for keeping up the sensitivity. So within the Agile Optics technology, we allow you to trade off between resolution and sensitivity of the system. So wide field imaging for your fastest throughput possible, the most sensitive. We have this unique 50 micron slit that allows for high throughput confocal imaging. It rejects quite a bit of background, gets you some of that confocality to be able to see your thick samples more sharply. And then we have two pinhole disks for either basic confocal imaging or high resolution confocal imaging. Moving on to the software. Our products are driven by MetaXpress. It's an integrated image acquisition and analysis tool set. We have a variety of canned application modules for turnkey assays, such as scoring cells for intensity of brightness or cell cycle. At the other end of the extreme, we have journals, which are macros, which are powerful tools that allow you to do nearly any type of image analysis and combine that image analysis with instrument control to make decisions on the fly. In between that, we have custom modules that give you a flexible tool that allows you to create an app module, create it once, and then only modify settings as required. We'll be talking about that a little bit. So the custom modules allow you to build your own application modules to do things like mitotic spindle and centrosome detection, label-free proliferation, finding objects and without stains. Not just doing something like neurite outgrowth, which is a CAN module, but finding punctures on the edge of neurite, finding puncture and counting those objects, or migration assays. But what we've added new to this tool is the 3D analysis package. There's been a desire and a problem of having these 3D assays working for assay development, but not knowing how to scale them up and analyze across multiple treatment wells. So currently, it's complex workflow involving third-party software, maybe importing and working with a bioinformatics expert. What we've added to the tool is to be able to do these 3D measurements. What you're seeing on the left is a spheroid and pseudo-colored. The objects are cells that are distant from the spheroid. As it moves, the flat side is on the glass surface, and the red cells are the furthest away from the spheroid. So that's a distance measurement that has been obtained from the sample. So we try to develop an integrated seamless acquisition analysis tool for doing 3D data for multiple well assays. We want to be able to quantify 3D structures with 3D measurements such as volume, intensity, and distance to remove analysis as a barrier to scaling up complex biology. And we wanted something that's compatible with wide field confocal data. And that is what we have developed. And so here is an example experiment that Oksana is going to talk to you much more about, where we're showing a control spheroid and a sporospore-treated spheroid. And this is not an individual cell. This is actually spheroids with hundreds of cells in that spheroid. And we are showing you that, Excuse me. Oh, it's not. I apologize. The movie's not playing. Okay, I apologize. So that volume, that green thing is a volume measurement, and there would have been a movie that played and showed it rotating. Oksana will have it in her deck.